Okay, why don't we get started? Hello, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. Please type any scientific questions in the Zoom question and answer box. If you mistakenly put it in the chat box, I'll redirect you to put it in the Q&A box. All attendees can prioritize questions by clicking on the thumbs up / like button. Questions will be responded to at the end of both presentations. Closed captioning is available. The link will be available in the Zoom chat box. You can cut and paste that in a separate web browser. If you're having any technical issues, you can make a comment in the Zoom chat box. With that, I'd like to turn over the proceedings to Dr. Lillian Kuo, who's a program director in the Cancer Immunology, Hematology, and Etiology branch in the Division of Cancer Biology. She's also, in my opinion, the great behind-the-scenes coordinator of all these great Cancer Moonshot seminars. Dr. Kuo.
Thanks, Alan. I'm here on behalf of our NCI Cancer Moonshot program planning team. Based on success and feedback from last year's seminars, this year we're highlighting the outstanding work of early career scientists or junior investigators supported through the Cancer Moonshot. Based on nominations from Cancer Moonshot investigators, these abstract-driven talks were selected. Today is the first of 3 of these special Cancer Moonshot seminars featuring 2 junior investigators paired together with complementary areas of research. We welcome you to follow and continue the discussion on Twitter using hashtag CMSSJI. To facilitate our discussion, we are honored to have Dr. Sarah Tajan to serve as our Cancer Moonshot moderator. Dr. Tajan is a pediatric oncologist and physician scientist at the Children's Hospital of Philadelphia and University of Pennsylvania School of Medicine. She's an internationally recognized expert in developing CAR T-cell therapies for pediatric ALL and AML. Thank you, Sarah. I'll hand it over to you.
Thanks so much, Dr. Kuo. And I'm delighted to be here today with our early career investigators and excited to introduce our first speaker, who is Dr. Rogelio Hernandez-Lopez, who's coming to us today from UCSF. Dr. Hernandez-Lopez obtained his Ph.D. in chemical physics at Harvard University, where he studied mechanisms of microtubule motors. He's currently a postdoc at UCSF in Wendell Lim's lab, focused on cellular immunotherapies for cancers. Rogelio has earned multiple nominations and early career investigator awards, and importantly, has been very involved in a lot of international nonprofit work to increase STEM education in Mexico and other countries. Dr. Hernandez-Lopez, we're really looking forward to your talk. Thank you.
Thank you so much for the introduction. I'm gonna start sharing here. Okay. Yeah. Can you see now my slides?
Yes, looks good.
Perfect. Yeah, I'm really excited to tell you today about our efforts to expand the repertoire of the cancer that can be treated with this transformative technology that is the engineering T cells for cancer targeting. So today, the work I'm going to present is in the context of using the approach of synthetic receptors into cytotoxic T cells to target cancer. And in particular, I'm going to talk about one challenge that is a major roadblock to expand this approach to other cancers, in particular solid tumors. And this is the problem of a specific recognition and control of the cytotoxic activity of T cells. And why is this a problem, and in particular the case of solid tumors, is that there is very, very few tumor-specific antigens. So far, the remarkable success that this approach has shown in the case of blood cancers, lymphoma, and leukemia, it's because targeting the CD19 antigen really only marks the tumor cells. And although that antigen is also present in other normal cells, killing those normal cells is not it doesn't threaten the health of the patient. It is manageable, but that is not the case of trying to use these antigens that are commonly expressed in solid tumors like HER2, EGFR, and GD2, because those antigens are also present in normal tissue, although at lower levels. So using the approach of engineering T cells against these antigens results in killing of both tumors, but also healthy cells, normal tissue. And as you may imagine, this could be a really big issue because, and there has been reports of toxicity. For example, here I'm showing you a microscopic experiment where anti-HER2 T cells have been put in co-cultures with cells that express either levels that represent tumors, but also the levels that you will find in normal tissue. And as you see, both types of cells get killed. So what we wanted to do was to create a new way of controlling the activity of the T cells, and in particular to have this sigmoidal response that is known as ultrasensitive response. So ideally, you want to have the full cytotoxic activity of these T cells only when they encounter tumor cells, but do not do anything when they encounter cells that express lower levels, those that you will find in normal tissue. So there is multiple ways in which you can create these ultrasensitive responses, but we decided to go with one that is widespread in biology, which is the use of the multi-step circuits. For example, one that is found in T cell biology and how the T cell sends the IL-2 is that an initial low affinity interaction with IL-2 using receptors triggers the expression of other receptor chains that form now a high affinity receptor that leads to the full IL-2 signaling and activation of these T cells. So we thought that using the synthetic biology approaches and synthetic receptors that are now available for T cell engineering, we could build something like that, but for the case of antigen density discrimination. So let me introduce very quickly this new class of synthetic receptors called SynNotch receptors. They were developed a few years ago in the lab of Wendell Lim by Leonardo Morsuta and Cole Roybal. And the way to think about these receptors is that they convert a binding event into a transcription activation event. So the way in which these receptors are created is that you replace the extracellular portion of Notch now for a recognition domain that could be the scFv targeting an antigen of your choice. And at the same time, you replace the intracellular portion of it for a transcription factor that could lead to the activation of orthogonal and very specific outputs. So in this way, you convert a novel environmental input, in this case, an antigen, into a transcription activation event. And these synthetic receptors have been used in a wide range of scenarios, and in particular for the cancer therapeutics in building what the so-called ANGATEs. But we use these receptors in a slightly different fashion. And we build these antigen density sensors using the synthetic Notch receptor, controlling the activity of the chimeric antigen receptors that ultimately are going to lead to the T cell activation and T cell killing, with the additional twist that the affinity of the synthetic Notch receptor is low, and that works as a high density filter, while the affinity of the chimeric antigen receptor is of high affinity to lead to the activation and potent killing activity of these T cells. And so the way we thought about these circuits is shown schematically here in this figure. If you have a synthetic Notch receptor that is low affinity and encounters target cells that have low expression of the antigen, Then, because the affinity is low, the frequency of the interaction between the antigen and the receptor is going to be low, leading to no CAR expression or very, very low CAR expression. In contrast, if these low affinity synthetic notch receptor encounter now targets that express very high levels of the antigen, which are the landmark of several cancers, that should lead to the full activation of the CAR expression. Once the CAR engages because it's of a high affinity, it will lead to the killing and expansion and the full signaling program of the T cells. So in order to test this idea, first we built a series of synthetic target lines that have different levels of an antigen. And we use as a test case the HER2, which is known to be overexpressing several cancer types like ovarian or breast cancer. And we also created lines that have lower levels representative of normal tissue. For example, here below, I'm showing a comparison of the expression levels of HER2 in a normal breast cell. And we sought out the goal to differentiate cells that have 100-fold difference in expression. If you can see here, we quantitate the number of molecules of HER2 present in cancer cell to be close to 10 to the 7 and normal tissue around 10 to the 4.7 or so. So this 100-fold difference in expression is a reasonable goal for full discrimination. And this is in agreement with a recent report showing that the levels of an array of normal tissues is indeed 100-fold difference compared to the levels that you will find of HER2 in cancer cells. So in order to build the circuits that I described earlier, we also made a series of antigen recognition domains that expand also around 100-fold difference in affinity, single point mutations or double mutations into the recognition site of this Herceptin antibody were introduced in order to achieve these different variants. So we built either the two-step circuit, synthetic notch receptor controlling the activity of the CAR, and we compare the activity of those circuit T cells with just expressing constitutively the CAR expression. And we found a remarkable difference in the behavior. We built series of these circuits. For example, here, the synthetic notch receptor of low affinity controlling the expression of a high affinity CAR, and also other variants that I will describe in a moment that, for example, the low affinity notch controlling the expression of a low affinity CAR to show that you have some room for tuning the behavior of these T cells. So these are the quantitation of flow cytometry experiments where we co-culture the target engineered cancer cells with the T cells and measure the number of dead cells after 3 days of co-culturing. And as you can see also here, the behavior of these synthetic circuit T cells is very different from what you will find if you use only a constitutive expression of a CAR, either of a high affinity or a low affinity. So we further investigated the circuit that had the highest ultrasensitivity and showed that the behavior of the circuit is robust and across multiple donors. And now I want to tell you a little bit more about the mechanism and why we think this strategy works. As I mentioned earlier, we thought about using the synthetic notch to control the CAR expression. And here I am showing you flow cytometry plots that have the CAR expression as a function of the density of the HER2 in the target line. So as you can see, only when you co-culture the T cells with targets that have higher levels of HER2, you get robust CAR expression. When you co-culture these T cells with targets that have low density, the expression is low. But the other very important component for the behavior of this approach is the control of T cell proliferation. We saw robust proliferation of the T cells when they were co-cultured with these targets that express high levels of HER2, but low levels of proliferation or the same level that what you will find with untransduced. When you co-culture these T cells with targets that don't have any antigen. So it's both the control of the CAR expression and the control of the T cell proliferation that lead to the overall sigmoidal response in the cytotoxic activity of these T cells. So earlier I mentioned that because in the circuits you have 2 points of control, at least in terms of the affinity, we build a series of circuits fixing the affinity of the synthetic notch and then modifying the affinity of the CAR, and show that you can build a series of these circuit T cells that have different behaviors. And actually, if you lower the affinity of the CAR, you can tune the threshold of activation of these T cells, although also the overall activity of the T cells, it's lower when you use the low affinity CAR. Not surprisingly, and this is also by design, if you increase the affinity of the synthetic notch to something that is a medium affinity, for example, then you lose the ultrasensitivity. And the way to think about this is that using a higher affinity synthetic notch, now you are also expressing the CAR molecules when you don't need those CAR molecules, which is the case on the low density target cells. So in addition of the flow cytometry experiments, we perform microscopy assays tracking the target cells over time when we co-culture these targets with T cells. And earlier I showed you that if you use CARs that are either of a high affinity or low affinity, you don't get any selectivity. But in the case of the circuit using the synthetic notch of a low affinity controlling the affinity, the CAR expression, you get robust selectivity. So in this case, this is a line that expresses high levels of HER2, ovarian cancer, and this is another cell that express low levels of HER2. Something that you may have noticed in the experiments that I showed you earlier is that the circuit T cells take longer for the full killing of the target population. And the reason for this is that the T cells need some time to build enough transcription activation and then CAR expression in order to get at the point where they activate the cytotoxic activity. So in this case, there is a compromise between the density discrimination and the killing speed, but nevertheless, this design allows this robust discrimination. So let me show you a few other assays. We also wonder what would happen in other environments, for example, 3D spheroids that resemble more microenvironments that could be found in tumors. So as a first set of experiments, what we did was build a line that normally forms spheroids, but this time to express high levels of HER2. This breast line expresses low level of HER2 on its own. So using these spheroids and co-culturing now with T cells in a matrix, we found similar results with the 2D cultures and the flow cytometry experiments, where there is a robust selectivity between the high density spheroids and the low density spheroids. Something that I think is going to be very important for this type of experiments and settings is that then you can start asking other type of questions that have to do with the composition of the tumor and also the spatial localization of other structures or tissues surrounding those tumors. In a first set of experiments, we co-culture these high density spheroids with arrays of low density spheroids, and then using the circuit T cells, we found that the killing in this case is very localized. So there is killing of low HER2 spheroids, but only the ones that are very, very close to the tumor. Other structures that are far apart from these spheroids that have high levels of HER2 are not getting killed. So finally, we did a series of 2 tumor mouse model xenografts in NSG, where one flank is implanted with a high HER2 tumor, the other flank is implanted with a low HER2 tumor. I show that after injecting these circuit T cells, the only tumor that gets killed is the high HER2 tumor. And that is consistent with finding a larger number of T cells that also have a higher levels of CAR expression in the high HER2 tumor. We did this type of assays and compared them with what will happen with either a low affinity CAR or high affinity CAR that, as I mentioned earlier, don't have any discrimination. In addition to the engineer target cell lines, we also tested these experiments using ovarian cancer or breast cancer and other cell lines that express a low levels of HER2, and showed that indeed the only tumors that are getting killed are the ones that express high levels of HER2. And that also correlates with a preferential localization of these T cells only in the tumors that express high levels of HER2. Finally, we think that this approach could be used to build antigen density sensors against any other antigen. And as a test case also, we use the same approach, but this time against EGFR, and compare what would happen if you use the circuit approach or only the T cells that express the constitutive CAR, and only the circuit approach gives you this robust discrimination. So today I told you about our strategy to now control the recognition and the activity of this T cell to only kill tumor cells that express high levels of an antigen and spare the ones, those cells that express low levels. With that, I'd like to thank the LIM Lab, Wendell Lim, Seth Gardner, Kale Saxila, and their labs, and all the people that collaborated in this work, and of course the IOTN network and the grants and funding. Thank you so much for your attention.
Thank you so much, Dr. Hernandez-Lopez. That was an amazing talk, and we're really looking forward to hearing some questions at the end of the session. I am now delighted to present our second speaker, who also is coming to us today from the Bay Area, Dr. Evan Weber, who earned his PhD in immunology at Northwestern University and is currently a postdoctoral fellow at Stanford University in Crystal Makel's lab there, where he's been studying CAR T-cell engineering and really done some interesting work on T-cell exhaustion mechanisms. Evan, we're really excited to hear your talk. Thank you.
Thank you very much, Sarah, for the introduction, and thank you to the organizers of this seminar series for the opportunity to speak to you today about some recent work from our lab that was published earlier this year on exhaustion in CAR T cells and one approach to mitigate this. So these are my disclosures. So T cell exhaustion is a phenomenon that occurs in settings of chronic virus or cancer, whereby CD8 positive T cells, instead of clearing the infection and basically becoming effector T cells and then eventually a highly polyfunctional subset of memory T cells, in the setting of this sort of chronic signaling, these T cells embark on an altered differentiation path whereby they lose the ability to kill and secrete cytokine, as well as other effector functions. And they upregulate inhibitory receptors on their cell surface like PD-1. And eventually these cells also manifest transcriptional and epigenetic dysregulation, which is mediated by upregulation of transcription factors like TOX, NR4A family transcription factors, and AP1 family transcription factors. And T cell exhaustion, we now know, also occurs in CAR T cells. And we know this from correlative data from patient samples, but we also have been able to study this in several preclinical models. And some of the earliest data on exhaustion in CAR T cells came from Crystal's group in a paper that was published in 2015. And the lead author, Adrian Long, showed first by these immunofluorescence images that the CAR protein can actually cluster in the absence of antigen. And so what you're seeing here is a CD19 targeting CAR that's really nicely diffusely expressed on the cell surface of these human CAR T cells, and a CAR targeting the ganglioside GD2, which is an antigen that's expressed on many solid tumors. And this CAR, as you can see, sort of creates this punctate-like pattern on the surface of CAR T cells. And this is actually antigen-independent CAR clustering. And what's kind of amazing about this finding is that this antigen-independent clustering leads to hallmarks of exhaustion. So these CAR T cells that express the GD2 CAR upregulate inhibitory receptors on the cell surface like PD-1, TIM3, and LAG3. And when you probe these cells functionally, here in a xenograft model where the tumor expresses both CD19 and GD2, the CD19 CAR T cells are able to really nicely control tumor growth in this experiment, but those targeting GD2 basically show no effect. We went on to leverage this GD2 targeting CAR and actually sort of supercharge it in a way to make it even more, to make it tonically signal even more intensely. And this was work published in 2019 from our lab, where we showed by using this tonically signaling exhaustion model, we could actually drive these CAR T cells to manifest epigenetic dysregulation similar to that which is observed in other exhaustion models like LCMV and syngeneic tumor models. And this is defined by tens of thousands of differentially accessible loci in the chromatin, and then also a lot of these differentially accessible regions contain motifs to transcription factors that are known to promote exhaustion. And so thinking about exhaustion through the lens of chronic CAR signaling, we reasoned that this signaling, sort of creates this vicious cycle where you have either tonic or tonic CAR signaling induced by CAR clustering in the absence of antigen, or antigen-induced CAR signaling leads to T cell dysfunction, which leads to a poor anti-tumor response. And this results in higher tumor burden, thus feeding this sort of chronic CAR signaling. And we asked whether we could use synthetic biology to break this cycle, thereby resting or disrupting CAR signaling and resting these CAR T cells down in a way that might induce functional reinvigoration and a more potent anti-tumor response. And so we reasoned that in order to disrupt CAR signaling, we would have to figure out a way to control CAR expression using synthetic biology. And so to achieve this, we collaborated with Tom Wanless at Stanford University, who developed this really nice destabilizing domain technology years ago. And we incorporated his technology as a way to finely tune and control CAR signaling. The way this works is you basically fuse one of these domains to the far C terminus of the CAR. At baseline, this CAR immediately gets degraded. It never makes it to the cell surface. But in the presence of a specific stabilizing small molecule drug, we could stabilize this domain, thereby stabilizing the entire protein fusion, and achieve dose-dependent regulatable surface CAR expression, as you can see here, using SHIELD-1 as our stabilizing drug and measuring CAR surface expression based on a flow cytometry assay. And so sort of merging these two concepts, first, this tonically signaling exhaustion model and destabilizing domain technology, we could create a model where we could really test this question in vitro and in vivo, whether resting CAR T cells by downregulating the CAR could result in functional reinvigoration. And that's sort of illustrated by this schematic here. In the presence of drug, we achieve CAR expression, which induces tonic signaling and exhaustion. In the absence of drug, the CAR is not expressed, and you can disrupt this tonic signaling. And so this is a sort of a model system that I'm going to be describing more about in the next several slides. But basically, we've sort of named some of these conditions based on the duration of tonic signaling that we promote using this regulatable system. And so always-on CAR T cells are basically exhausted T cells that constitutively express this CAR for 15 days in vitro. Always-off CAR T cells are your sort of healthy controls. And then we have two rested conditions. One condition rested between days 7 and 15, and sort of a shorter rested condition where rest starts at day 11. And then finally, we have an always-on plus anti-PD-1 control, mostly because these T cells, they actually do express both PD-1 and PD-L1 on their surface, thereby implicating the signaling axis in our model, but also as a benchmark to which we could compare the effects of rest. And one of the first things we noticed is that in contrast to these sort of exhausted always-on CAR T cells, those that were rested, again, for as short as 4 days, demonstrate this profound downregulation of inhibitory receptor expression on their surface. And I think we expected some changes in this model, but we were actually pretty excited by the degree to which these inhibitory receptors were downregulated. And this was concomitant with changes to T cell differentiation phenotype. So in these cells that were rested, they actually begin to resemble the phenotype of the always-off healthy controls. So more sort of memory-like phenotype. We see higher CCR7 expression, higher CD62 ligand expression, IL-7 receptor, and many others, in contrast to the exhausted CAR T cells, which look much more effector or terminal effector-like. Now, the phenotyping was very interesting to us, but really we wanted to know whether these periods of rest, or this period of rest, was actually inducing functional reinvigoration in these cells. So in order to probe functionality in this system, regardless of the way in which these cells were treated during the culture period, so whether the CAR was on or off, 16 hours prior to tumor challenge, we reintroduced this stabilizing drug into our culture system so that we could normalize CAR expression across groups. And when we probed these cells functionally, we saw some really interesting results. So on the left here, I'm showing an IncuCyte assay in which we can track tumor cell GFP over time. And we see that in contrast to the exhausted CAR T cells, which are unable to control tumor growth, those that were either had never tonically signaled, or those that were rested for 7 or 4 days were able to mediate robust tumor killing. And this effect also extended to cytokine secretion. So here I'm showing IL-2, and in contrast to the exhausted CAR T cells or exhausted CAR T cells cultured in the presence of anti-PD-1, those that were rested are able to secrete much more IL-2 in response to GD2 positive tumor. And so we asked, well, this is really great. I mean, we know these cells look better, they function better. But what's really happening at the level of transcription? And so to do this, we conducted bulk RNA sequencing on day 15 of our different sort of conditions here. And we plotted these global sort of transcriptional profiles using a principal component analysis. And what we see is that basically all of the exhausted CAR T cells, whether they were treated with anti-PD-1 or not, end up on one side of PC1. And the rested CAR T cells and healthy CAR T cell conditions from 3 separate donors end up on the other side of PC1, really telling us that there's sort of this global transcriptional reprogramming occurring during rest. And when we dived a little bit deeper in the genes that define principal component 1 in this experiment, we see sort of 2 gene clusters emerge. So there are those that are highly upregulated in the always-on exhausted CAR T cells and downregulated in the healthy always-off and rested CAR T cell conditions. And these include your sort of inhibitory receptors like PD-1 and LAG3 and CD39, but also sort of major transcription factors like BADF and NR4A family transcription factors that have been implicated in exhaustion promotion in other settings, or in other models. And then on the bottom here, we sort of see the opposite trend, genes that are downregulated in exhausted T cells in this assay and upregulated in rested conditions. And these include your sort of memory or quiescence markers. And importantly, we decided to look at sort of the clonotypic diversity within each of these samples. And we found almost identical clonal diversity across all groups. And this suggested to us that the changes that we were seeing likely were not due to sort of outgrowth of a minor subpopulation of cells that were maybe exhaustion resistant or had some sort of differential sort of effect to the rest. So basically this data suggests that these changes are sort of occur at the population-wide level and not this small outgrowth of an exhaustion resistant subset of cells. And finally, this transcriptional data really suggested to us that we might actually be seeing epigenetic changes in these cells as well. Although I should say that in other models where checkpoint blockade is tested or other models from our group where we can mitigate exhaustion in CAR T cells, we never actually observed epigenetic reprogramming. And so we asked whether rest could induce changes to the epigenome of exhausted CAR T cells. And so to do this, we collaborated with Howard Chang, Kevin Parker, and Ansu Satpathy, and we basically conducted a TAC-seq at various time points in our culture system. And we found really dramatic changes in chromatin accessibility across groups. So we see in rested cells, either those rested between day 7 and 11 or day 11 and day 15, we see over 10,000 changes to chromatin, either closed peaks or open peaks in these conditions. And similar to our RNA sequencing data, when you plot these conditions using principal component analysis, we see sort of a similar trend emerge where the exhausted CAR T cells, epigenetically, the always-on exhausted CAR T cells resemble those that were cultured in the presence of anti-PD-1, again, sort of confirming some of the data that had come out of John Wary's lab in 2016 around checkpoint blockade not really affecting the epigenome. Whereas those CAR T cells that were rested for as short as 4 days really moved to the other side of principal component 1 on this plot, similar to our sort of healthy controls. And so we asked whether this epigenetic remodeling that occurs during rest is actually required for the restored functionality that we observe in these cells. And to answer this question, we basically screened a couple of epigenetic drugs and cultured CAR T cells that were being rested in the presence of these drugs and asked whether we could disrupt this functional reinvigoration. And so the drug that we ended up sort of exploring in more detail is one that's called tazemetostat. This is a clinically available drug that targets EZH2, which is a histone methyltransferase. And what we found was that in contrast to the exhausted always-on or healthy always-off controls, where EZH2 inhibition really negligibly affects these cells, in the rested CAR T cells, we see this dose-dependent response, where as you increase EZH2 inhibition, you basically are able to blunt this restored functionality in IL-2 secretion in response to tumor in these rested cells. And this effect also extended to our killing assay, where we track GFP, tumor GFP fluorescence over time. And again, we see that EZH2 inhibition doesn't really greatly affect the always-on or always-off conditions, but those rested CAR T cells that were treated in the presence of EZH2 now look much more like the always-on CAR T cells, in contrast to the rested CAR T cells treated with vehicle, which again, similar to previous experiments, demonstrate this functional reinvigoration. And when we looked at this sort of H3K27 trimethylation landscape, and this is the modification that EZH2 is known to make, we see that a lot of the changes that occur in rested CAR T cells that are treated with this EZH2 inhibitor occur at or near gene loci that are associated with exhaustion. And these include NR4A family transcription factors, DNMT3A, and many AP1 family transcription factors, indicating that during rest, EZH2 might be responsible for turning off these sort of exhaustion-associated genes. And it, I think more broadly, also tells us that, that epigenetic reprogramming is really essential for functional reinvigoration of exhausted CAR T cells. Now, we also began to think about alternative strategies to induce rest. And in 2019, we published a study showing that we could actually use this clinically available drug called dasatinib, which is a Src kinase inhibitor, to potently disrupt CAR signaling, basically turn off the CAR by targeting LCK in these T cells, and that this effect was reversible, so that when you wash away the dasatinib, these CAR T cells are able to regain functionality perfectly. And so we asked whether we could also leverage dasatinib as a way to rest CAR T cells. And so first, to test this, we essentially cultured our CAR T cells ex vivo in the presence of dasatinib or in the absence of dasatinib, and then tested their functionality. And here we're using a GD2 targeting CAR with a 4-1BB co-stimulatory domain. And we find that culturing these CAR T cells, which tonically signal, in the presence of dasatinib for just 1 week, we're able to preserve this really nice sort of central memory phenotype in these CAR T cells. And when we wash away the dasatinib and infuse these CAR T cells into tumor-bearing mice, we find that those that were treated with dasatinib are able to control tumor growth much more effectively than those that were not. And now, sort of transitioning the strategy to our system of rest. Now, basically, we're using this tonically signaling CAR as a way to model exhaustion, as I had shown in previous slides. And we see that similar to regulating CAR surface expression with a destabilizing domain, by simply culturing these exhausted CAR T cells in the presence of dasatinib, we can reverse a lot of the hallmarks of exhaustion. So here we see really incredible reduction of PD-1, LAG3, and TIM3 expression, as well as this marker CD39, which is actually, for us at least, a very nice readout of T cell dysfunction. And concomitant with these changes in exhaustion phenotype, surface phenotype, we see pretty profound changes to the expression of many transcription factors associated with exhaustion or memory. So in contrast to these vehicle-treated exhausted CAR T cells, which express high levels of TBET and TOX, those that are rested with dasatinib express much lower levels of these exhausted associated transcription factors. And inversely, we see that the longer that you rest CAR T cells in the presence of dasatinib, the more LEF1 and TCF1, these memory-associated transcription factors, you see. And then finally, we observed really profound functional reinvigoration in these dasatinib-treated CAR T cells, both by killing assay in which we track tumor GFP fluorescence over time. And we also see this reinvigoration of the capacity to secrete cytokine in response to tumor. And actually this, at the level of cytokine, we see that those CAR T cells which are rested for longer with dasatinib actually have a greater degree of functional reinvigoration than those that were rested for a shorter amount of time. Now, finally, we asked whether we could use dasatinib in vivo to rest CAR T cells and prolong anti-tumor responses. And so here we're using, I'm going to show a couple of dasatinib insensitive xenograft models. The first being a NALM-6 model, and the CAR T cells we're using here are actually non-tonically signaling CD19 targeting CAR T cells. And we found that after 2 short pulses of dasatinib, we see that these mice, which were dosed with dasatinib, demonstrate much better tumor control over time in comparison to mice treated with vehicle or mice treated with dasatinib every other day. So again, this sort of gets back at the question of duration, where the longer you rest these CAR T cells, the more effective this maneuver is for tumor control. And again, these mice that were treated with this dasatinib pulse also had a survival advantage. We also used a solid tumor model here, 143B osteosarcoma, and the CAR T cells we're using are GD2 targeting CAR. And we see in this setting as well, intermittent dasatinib pulsing is able to sort of prolong the anti-tumor response and sort of make a more durable response in this very aggressive model. And when we look at these tumors after one pulse, so either a short 3-day pulse of dasatinib or a longer 7-day pulse of dasatinib, and we take out the tumor infiltrating CAR T cells, we see that those mice which were dosed with dasatinib, their CAR T cells display a more memory-like phenotype, as you can see here, based on CD62 ligand and CD45 RA expression. And when you stimulate these tumor infiltrating CAR T cells ex vivo, we see that those that were treated with dasatinib are able to they express more CD107A than those that were treated with vehicle.
And I'm sorry to interrupt you, but we're going to need to wrap up.
Yeah. No, this is the last slide. Thanks, Sarah. So in summary, we really wanted to explore the biology of CAR T cell exhaustion in these models, and we basically came across this finding where by resting or transiently inhibiting CAR signaling, we could induce restored function, memory-like phenotype, transcriptional reprogramming, and epigenetic remodeling. And we think that this is a translatable maneuver that could be incorporated now in many regulatable CAR T cell systems. We think that this work challenges the notion that exhaustion is a completely irreversible cell state, and that this sort of lays the foundation for future work targeting the exhaustion-associated epigenome as a way to improve efficacy. And with that, I simply want to acknowledge everyone who was involved with this study. Really a huge team effort, and led by also by Crystal, Elena Satia, Satio, Rachel Lynn, and Kevin Parker, our funding sources, especially Cancer Moonshot. And I thank you all for your attention and happy to answer any questions. Thanks so much.
Thanks, Dr. Weber. That was really incredible. So we'll open up the floor now for questions and answers and discussion with our two early career investigators. I really want to congratulate both of them on their amazing work and accomplishments in fellowship and instructorship and their high profile publications. Dr. Hernandez-Lopez is going to be an assistant professor at Stanford starting in 2022 in the departments of bioengineering and genetics. And Dr. Weber has also just accepted a faculty position. So congratulations to you both. Great. So we'll get started with the Q&As. So the first question is from Dr. Susan McCarthy. And this is a question for Dr. Weber. She would like to know, are the always off cells getting tonic survival cells, or I guess signals from another receptor, or are they tonic independent?
Yeah, thanks, Susan, for your question. So the always off cells, basically in order to generate CAR T cells, we have to activate these cells with CD3, anti-CD3, anti-CD28 beads. And so I think, and also these human CAR T cells are being cultured ex vivo in the presence of IL-2. So even though they're not receiving tonic signaling through the CAR, they are getting some survival signals through the IL-2 that they're sitting in, as well as that activation on day 0 in our in vitro experiments. So I hope that makes sense.
And our next question is from Sarah Elizabeth Siegler. And I think this is a question a little bit more for Dr. Hernandez-Lopez. Her question is, relapse by mechanisms that involve antigen modulation have been observed in blood cancers following CAR T cells. Do these T cells adapt in such circumstances? And if so, how?
Yeah, that's also a very good question. I think there is still it remains to be seen whether the mechanisms of antigen modulation and downregulation that have been observed in the case of blood cancer also applied in the case of solid tumors. The case that I show is the one of HER2, which is massively overexpressed in several cancer. And it seems that, like, although after hitting the cells with T cells, like there is downregulation of the antigen, there is still enough antigen around for the T cells to still kill those cancer cells. I think in the case of HER2, there is also reports of lack of efficacy, but only against antibodies or antibody drug conjugates. So I don't think it has been shown that there is escape using CAR T cells. That's something that still needs to be explored.
Thank you. And our next question is from Dennis Dogbe, who's asking what antigen concentration gradient defines whether low or high. I think this is a question also for Dr. Hernandez-Lopez.
Yeah, that's a very good question. In general, in the clinic, there is the traditional immunohistochemistry, that is used to detect the amounts of a particular antigen that is present. In the case of HER2, there is also and other antigens, there is also tools like fluorescence in situ hybridization that measures the amounts of RNA. But then later, there is like several ways in which pathologists and other people have decided to call either the intensity of the fluorescence high or low or medium. So I think there's still room for making the quantitations kind of like more accurate, in terms of like defining numbers and how many molecules defines a certain degree of antigen expression. In the case of the C19, and I know work from the Makel lab has defined, like what's the threshold for what's the lowest amount of the C19 antigen that the T cells can detect. But that has been done with flow cytometry, and also the quantitation that I performed of the antigen levels was done by flow cytometry, but still there is room for improvement there.
And our next question is from anonymous attendee number 1 for the second talk. So for Dr. Weber, do the CAR T cells proliferate during the rest period, and is proliferation required or not required for restoring T cell function?
Yeah, that's a great question. So we have a little bit of data addressing that in our manuscript. It's actually buried in the data supplement. But in vitro, during rest, either with using our destabilizing domain method or using dasatinib, we actually do see a small increase in proliferation of CD8 positive CAR T cells. And in some ways, this is paradoxical, because when you remove antigen from T cells, typically after a certain amount of time, right, the T cell population will contract. And so, but during this very short rest period, we saw a boost in proliferation, which was surprising to us. We don't know, we haven't tested whether specifically that this proliferative burst is responsible for all of the restored functionality that we see in our functional assays. We also haven't tested this carefully in vivo yet, but it's definitely something we're thinking about, and potentially something we can try to leverage if this is indeed a really important mechanism for functional reinvigoration of exhausted CAR T cells.
Great, thank you. And then our next question from anonymous attendee number 2 for Dr. Hernandez-Lopez. What about HER2 expression in cancers, and do cancer cells also display a spectrum of HER2 expression?
Yeah, that's also a very good question. Although I describe kind of like the mean values of the expression of high, intermediate, low, there is cases of heterogeneity in the expression levels. In fact, the most challenging cases in breast cancer and other HER2 positive cancers are those that are heterogeneous in the expression. And that's something like a problem that I'm interested in, and other people are interested in, like how you tune the activity of these T cells to be able to deal with heterogeneous samples. And it could be either by using combination of antigens, no, or other ways in which you tune the durability of the CAR expression so that it can deal with a heterogeneous sample. No? But yeah, that's also a very good question.
Okay, great. And then a question for Dr. Weber from Brian Koss. Who looked into metabolic changes in the different rest, exhaustion, and the plus minus drug combination conditions?
Yeah, thanks for that question, Brian. That's it's a really good point, and it's something that we considered early on in this project. So we did some initial studies around metabolism, and we did see some changes, and it sort of it corroborates some of the phenotypic data that we observed in rested cells. So at a metabolic level, it did look like these rested CAR T cells resembled more of a sort of quiescent or memory-like phenotype. But we didn't look that carefully, and we didn't want to convolute our story too much with an additional sort of angle. So I think we'll save the metabolic stuff for later, but that could also certainly play into some of the sort of reinvigorating effects we see after rest.
Great. Then I have a question actually for both of you from anonymous attendee number 3, and a question I think I'm also interested in from a translational perspective. Both speakers described exciting approaches to tune CAR T cells. How would you envision future applications of these approaches towards the development of universal or off-the-shelf technologies? And this is with the caveat of this is a brainstorming question, not necessarily a technical question.
Rogelio, you want me to take this one first?
Yes, go ahead.
Yeah, so that's a good question. I mean, I think the most obvious approach that I've considered so far for translating this idea of rest to the clinic in sort of a more universal way is by combining dasatinib treatment with CAR T cells. And dasatinib is an already FDA-approved drug for the treatment of Philadelphia chromosome positive CML and ALL patients, and is pretty efficacious. And so potentially we might be able to observe synergy between CAR anti-tumor killing, CAR T cell anti-tumor killing, and dasatinib and the effect of dasatinib on tumors. And so, this idea of pulsing dasatinib specifically in that setting is really appealing. And it could be one that could be applied across, it's almost like CAR independent. It doesn't matter what you're targeting on the tumor or what the co-stimulatory domain is. We think that this approach is universal in that sense. And so we hope to be able to test this clinically in the near future. And we think it could be also a potentially safe way to test this hypothesis in people.
Rogelio, do you have any thoughts?
Yes, I think in the case of the synthetic circuits and in particular of antigen density sensors, the way to move this forward, and in particular for off the shelf, would be to explore. I mean, there's many advances in genome engineering that hopefully are going to lead to using these circuits perhaps in other cell types, NKs, or editing the TCR in T cells. So I think the synthetic biology field, the genome engineering field are going to come together to allow using this strategy for different types of cancer. I think what we show in terms of like the robustness of the activity and how you control using the synthetic receptors could be applied for essentially any type of cell, provided that you have this targeting, these changes in affinity, and yeah, I know that there is several people working, for example, in humanizing SynNotch, and using receptors that do not present any immunogenicity, which is also going to be important for the clinical application of this approach.
Thank you both. I think we are unfortunately at the top of the hour right now, so I want to really thank and congratulate our wonderful early career investigators again for presenting their exciting work with us today, and thank the audience for their attention. I'll hand this back over to Dr. Kuo to close us out. Thank you.
Yes, thank you. Thanks so much to both the speakers and Dr. Tazian for a wonderful discussion. And just a reminder, 2 weeks from now, we have a very special Cancer Moonshot seminar on the Blue Ribbon Panel progress, the Blue Ribbon Panel report anniversary, reflections, progress, and potential of the Cancer Moonshot, featuring Elena Martinez, Liz Jaffe, Dina Singer, Ned Sharpless, and Tyler Jack. So please register and tune in. And thanks everyone and have a great day.